Biology 1308, March 23rd lecture. We are starting with page 155. This is where we left off before on recessive disorders. Now, recessive disorders are the most common genetic disorders. Now, the other thing about recessive disorders is they are often involving what's called carriers. Now, the carriers are typically the ones that are homozygous recessive. That's what we learned last week regarding two small case letters. For example, little r would be a recessive homozygous, and those are the ones that are considered carriers. Now, on our next slide, we're going to see an example on page 155. Now, this is the example that we ended with that we're going to continue with. Here I see two parents that are actually carriers because they have a phenotype which is normal, but genetically they are genotype in terms of a heterozygous, capital D, little d, for being a carrier. In this example, it says I have a heterozygous mom and a heterozygous dad, and in both cases, it produces, and they're both carriers, it produces a 50% chance of having a kid that's also a carrier. It produces a 25% chance that it's going to be a normal homozygous dominant, capital D, capital D. It also produces a 25% chance that it's going to be deaf with a homozygous recessive. Now, in this example, it does not mean that if I have 4 kids, this is my storyline. Rather, it means per child. So there is a 25% chance that that one child, or the per child, that one child is normal hearing. There's a 50% chance that that child is going to be a carrier. And there's another 25% chance per child that that child will be deaf. So this is a recessive dominant, it's a recessive disorder. So this is a recessive disorder, page 155. I do not care for the example, so on page 155, in a previous lecture, I just simply said you need to be aware of recessive disorders and dominant disorders from the chart. I do not care for the example. Okay. Now, this is a different story on page 156. On this one, this is a dominant disorder. How do I know? Because I'm actually looking at a capital D here. So a dominant disorder simply means as long as I have a capital D, I'm going to have this disease. In this case, achondroplasia, which deals with dwarfism. So achondroplasia dealing with dwarfism. As long as I have 2 little d's, it's considered normal. Now notice in this example, I end up with having a 50% chance. So dominant diseases can be a little bit more destructive because it's now a 50% chance of actually having a child with dwarfism. Okay, that was page 156. Okay, now don't care, don't care, don't care, don't care. Now this one I do care. This is page 157, genetic testing. Now, in order to discover whether there is a genetic problem prior to having your kid, so inside mommy's tummy, we perform 2 possible tests. The first test is amniocentesis. And in amniocentesis, the issue is where they will draw a long needle, take the fluid of the amniotic fluid, that's the fluid surrounding the baby when they're inside mommy's tummy. That amniocentesis needle will actually draw the amniotic fluid, and then they do a karyotype from that. So the cells that they harvest, they do a karyotype. Very similar to amniocentesis, on the next slide, is something called chorionic villus sampling. This is where they take a sample from the chorionic villus, which is part of the placenta. It's the baby side of the placenta. So they do what's called a CVS, chorionic villus sampling. And they draw the fluid, which is really the blood from that part of the placenta, and then again they make a karyotype from it, and then they can do other tests as well. So we have 2 tests to be aware of for genetic testing, amniocentesis and chorionic villus sampling. Okay, next slide. Here's a sample of one. So here's a picture of one being done. Next slide. Don't care. Now this slide I do care. This is page 158. Now this slide is unique. It says Mendel's two laws, and we've actually looked at Mendel's two laws. One was law of independent assortment, and that one had to do with characteristics. The other one's law of segregation, which dealt with traits. Now it turns out that in addition to Mendel's laws, there are other variations that can occur as well. So it's not just Mendel's law only that counts. There are other possibilities as well, and they're called variations. So starting on page 158, we're going to see our very first one. Our first variation is incomplete dominance. Now this slide doesn't do justice, so we're going to see a slide that does. So this is called incomplete dominance. The slide that's going to help you is this one on page 158. Now this slide shows you a red flower, which is a dominant, and then it shows you a white flower, which is recessive. Now previously what Mendel discovered with his purple and white was that purple was dominant over white. And that's why we ended up getting more purple flowers instead of white flowers. Well, now we have a different story. This is called incomplete dominance. So it's not red in this example over white or white over red, but it's actually a blend. And that's why we end up getting not just red as dominant or white as recessive, but we end up getting pink. So incomplete dominance would work this way. And notice we have a red, we have a 50% chance of pink, and we have a 25% chance of white. So this is page 158, incomplete dominance. Next slide. Don't care, don't care, don't care. Now this slide I do care. This is page 159. Now this deals with the blood groups, A, B, O, and AB blood group. Now your blood group is based upon a concept called codominance. So in an exam setting, you need to keep in mind that your blood type, whether it be A, B, AB, or O, the blood grouping is done based upon codominance. Okay, so codominance. And here we have 3 alleles, all based on a single gene. And that's all you need to know for the test. Don't care, don't care, don't care, don't care, don't care, don't care, don't care. Now this slide I do care. This is now a definition slide. The next category is pleiotropy. You will need to know the definition. So pleiotropy is when one gene influences several characteristics. So one gene affecting several characteristics. And the best example is sickle cell anemia or sickle cell disease. Here, one gene has many influences. So influences on several characteristics. Next slide. Don't care. Don't care. Now the next term. Now this one is called polygenic. Notice the definition. It says it's an additive effect of 2 or more genes, but on a single characteristic. So this is totally the opposite of pleiotropy. So totally opposite of pleiotropy. So this time with polygenic inheritance, 2 or more genes, but having an effect on a single phenotypic characteristic. The example that the book uses, and you'll likely see on a test, is height. So height is polygenic, and that's what they'll ask you on a test. So height is polygenic. It is not just one gene that determines the height. It's several genes affecting a single characteristic, like in this example, height, and that's called polygenic. Totally opposite of pleiotropy. Okay, next slide. Here's an example showing you how height has a lot of variations, but that's because it takes 2 or more genes to affect one characteristic called height. Next slide. Don't care, don't care, don't care. Okay, I'm now on page 161. Now 161 deals with what's called epigenetics and how it plays a role in the environment. Now epigenetics deals with how genes are affected by the environment. And that's that first bullet right there. How genes are affecting the characteristics, how genes are affecting the environment, and vice versa. The last bullet is of value to us. Spending time with identical twins will convince anyone that the environment, and not just genes, affects a person's traits. So you can have somebody living on the East Coast, somebody living on the West Coast, and because the environment is different, their outcome of their genes will be different. Here's an example. They're identical twins, but they have a variation in the way they look, their personality probably because the environment affects it. So the environment does affect the genetics, and that's called epigenetics. Okay. Here's another slide which says basically the same thing. I'll read the second paragraph. In recent years, however, biologists have begun to recognize the importance of epigenetic inheritance, the transmission of traits by mechanisms not directly involving the DNA. So the environment can affect the DNA. Next slide. Okay, don't care. Don't care. All right. Now, this one's important. It is now page 162. So we're switching over to page 162. Make sure you draw a line before we start this slide. Now, this one says chromosomal basis of inheritance. And this is a word we actually looked at last time. And that word was locus or loci. Remember, that is where the gene is located on the chromosome. So where the gene is located on the chromosome is a locus. So it does matter where the gene is. Next slide. All right, don't care for this one because we've already talked about it. We have law of segregation, which deals with traits, and the law of independent assortment that deals with the characteristics. And yes, they're all related to meiosis. Next slide. This is a big, huge slide, and notice again, we're talking about meiosis or meiosis, and that vastly affects the outcome. Now remember what I said for the test purposes, that if we have a homozygous dominant for both traits, in this case it's for both round and yellow, which makes it a dihybrid. Here we have wrinkled and green as a homozygous, again a dihybrid, and that results in a 9-3-3-1 breakdown. So remember that from before, a 9-3-3-1 breakdown. That's the ratio that you're likely to see when you have a homozygous dominant for two traits and a recessive dominant or recessive traits for two traits as well. In this case, a dihybrid. Next slide. Okay, now the next slide we care for is linked genes. Now linked genes are because the genes are so close to each other on the same chromosome. So linked genes are ones where the genes are so close to each other on the same chromosome. Now what does that mean if it's so close to each other? It means that they're likely to be inherited together, even with the crossing over story that we saw before. So they're so close that they're linked to each other. So if you get one characteristic, you're likely to get the other one as well. And the example I gave previously that I'm now bringing back is blonde hair, blue eyes. So if hair color is blonde and the blue eyes for the eye color, so we have two characteristics so close to each other, they're now linked. So eye color and height would go together. So that's called linked genes. Next slide. Okay, this one I don't care for. We've done this one previous. We already know that X and Y make a male and two X's make a female, and we've already seen that. We also know that the first 22 are called autosome chromosomes, and then the 23rd set would be called the sex chromosome, previously seen last week. Okay, don't care. Don't care. Don't care. Okay, this one I do care, page 163. Here we have a title called Sex-Linked Genes. In this case, it's found mostly on the X chromosome. Now what that means for us is that there are certain diseases that are linked to the X chromosome. That means that if you're an XX, remember one of the X's for a female comes from the mom, the other X would come from the dad. So women can be certain carriers for diseases if it's on one of the X's that came from the dad. All right, next slide. So we'll see more on these sex linked in a moment. Okay. Now on this slide, we see diseases that are linked as sex-linked. This is a value for the test. So this slide you do want to make a note of. So we have red-green color blindness, which goes with sex-linked genes. We have hemophilia that goes with sex-linked genes. And we have some types of muscular dystrophy that also go with sex-linked genes. Now historically, the two that are important are often the red-green color blindness and hemophilia. The textbook is going to give you an example of a royalty. The British royalty, for example, actually has a link with the Russian loyalty. Back then, the royalty of Russia and the royalty of England. And there is a connection that deals with hemophilia, and we'll see that in a moment. Here's the color blindness story. I don't really care too much about that. Here's an example of color blindness, and this relates to page 163. So if you are colorblind for red and green, then you would have trouble seeing the number 7 that you see on the slide. Next slide. Don't care for this one. I do care for this one. And this one deals with color blindness, and this one is definitely worth going over on page 164. So page 164. We're going to start with the figure A here in the corner. And here we see a normal female. So a normal female because of the XX and the capital N, normal female. Here we have a male that is XY, letting me know it's a male, but we see that it's also a colorblind individual because of that Y there and that little n there. So we have a normal female with a colorblind male. Now, when they have kids, here are the possibilities. Notice these over here are both females. So a female, a female, female, female. But this color shading here with the legend indicates they are carriers. And then here we have two normal males. Now what's going to happen is the parents were a normal female and a male that has color blindness, but they end up having kids. They all look normal, but the females are all carriers. So what happens if one of these females actually gets married with a normal male? So this is the next generation, and I'm taking one of these boxes and saying that now this is the second generation. And here I have a female with a normal male, and the female is a carrier, and look what happens. We end up getting a normal female, a normal male. We do get up a female here just like the mom. So the mom was a carrier, and this one child as a female is a carrier. And then we have a male here just like not the dad, but the grandfather. So remember I said that sometimes it skips generations. Well, in this example, it's a perfect example. Here's the grandfather that's colorblind, and now they end up getting a boy that's also colorblind. Dad didn't have it, but grandpa did. And that's why now it's a male colorblind. Now if I go further, and let's say I have just like before, I've got a carrier female, but instead of having a normal male, they get married to a male that is colorblind, just like this previous generation. So now we have a female that's a carrier and a male that has color blindness, and now look what the odds are. Very high. They're going to have one female chance of having a carrier, one that will have color blindness as a female, one normal male, and one male that is also colorblind. So as you can see, it can skip generations. And it's also where the females tend to be what? The females tend to be carriers. Because in a sex-linked, the females end up being carriers typically. Okay, that's important for a test. So the females end up being a carrier for the disease. Next slide. Okay, don't care, don't care, don't care. So we've already dealt with color blindness. Now we're going to deal with hemophilia. Now this slide talks about the British royalty, so I'm going to skip all of this writing here and go straight to page 164's little pedigree here. Here we have a pedigree showing the British royalty marrying someone from Russia, and then we have a child that has hemophilia, but as you can see, because it's sex-linked, look what happened. All the females were carriers for it. So Queen Victoria, Alice, Alexandra, these were all females, carriers, and then we had a boy that actually has hemophilia. So there is a relationship between Prince Henry and the other brother prince there that actually does have a chance of having hemophilia in their family. Okay, next slide. Okay, that takes me to this slide at the end of the chapter on page 165, simply letting us know that regardless of what kind of dog you have at home, it's originating from a wolf. So dogs are basically originating from a wolf, and that's their evolutionary tree. Page 165. Okay, next slide. Don't care. Don't care. Don't care. Don't care. Here's a picture that I would find worthy on page 166, explaining pleiotropy a little bit better, where you have a single gene but multiple outcomes or multiple characteristics coming from one gene. That's pleiotropy. Then this was polygenic inheritance. Remember, height was the example, where it takes multiple genes to affect a single thing. Now, I don't know why the author calls it a single trait. That's not really the way to say it. It's a single characteristic, in this case, height. Height is a characteristic, not really a trait, so that's misnamed here. So, height. Next slide. Don't care. Don't care. Don't care. Here's a repeat of what we did. Don't care. Don't care. Now, that takes care of our slide for the remainder of chapter 9.